Hey guys, welcome to AP Biology, and we're going to do lecture 1.5, which is carbon and the role of macromolecules. Here we go. Okay, so first of all, oops, what's going on? Okay, there we go. Sorry about that. We're going to be talking about biological macromolecules in this particular lecture, and there are four categories of biological molecules that we need to know about. We have carbohydrates, lipids, proteins, and of course, nucleic acids. And we're going to talk about the first two today, carbohydrates and lipids, in this presentation. So, first of all, when we talk about carbohydrates, we're talking about the fuel that your body basically runs on. Now, this is short term energy. We have fats, i.e., lipids, for long term energy storage, but carbohydrates are what our body uses very, very quickly in order to get the energy that we need to survive. Okay, so as I say, carbohydrates are short term energy storage. They contain three elements: carbon, hydrogen, and oxygen in a 1 to 2 to 1 molar ratio. So as you can see up here, the empirical formula for a carbohydrate is CH2O, where N is equal to the number of carbon atoms. So if I have 6 carbon atoms, I'm going to have 12 hydrogens and 6 oxygens, a 1 to 2 to 1 ratio. And again, carbohydrates can be monosaccharides, disaccharides, or polysaccharides. Remember, saccharides means sugar or sweet. Okay, so that's what they are. So our monosaccharides are things like glucose, fructose, galactose, deoxyribose, which is the sugar we find in DNA, and ribose, which is the sugar we find in RNA. And there are smaller ones and bigger ones and so on and so on. But if you counted up all the carbons and the oxygens, you would come up with a 1 to 2 to 1 molar ratio in these monosaccharides. And monosaccharides can be 3 to 7 carbons long, ring shaped, or linear. You throw a linear monosaccharide into a solution, i.e., water, it will spontaneously form into a ring shape because that's more stable in a watery environment. And these monosaccharides are also the building blocks for larger molecules such as cellulose, which is wood and what paper is made out of, and things like that. Yes, wood and paper are made out of sugar molecules. Okay, and even some forms of storage energy in the forms of glycogen, which is the polysaccharide that is stored in our muscle cells and things. Okay, so these are all examples of isomers. What's an isomer, you say? An isomer is basically where molecules had the same chemical formula, but they have a different structural formula. So if we look at these triose sugars, their formula is C3H6O3 for both of these two right here on the left-hand side. Same chemical formula, but they are put together slightly differently. The same with the pentose sugar, C5H10O5, and for the hexose sugar, C6H12O6. They're all put together slightly differently. And remember, structure determines function. So even though they have the same chemical formula, because they are built slightly differently, they will have slightly different functions and how they are broken down, how they're utilized, what they're for, so on and so on. Okay, so the most important one we like to think about is glucose, and you've all heard of glucose. It's a very important monosaccharide. Basically, our body, during the process of digestion, when we take in large carbohydrates, things like starch and disaccharides, even, we're trying to break our body through the process of digestion is trying to turn those large complex polysaccharides into glucose monomers, which can now be transported across cell membranes and into the cell and eventually to the mitochondria, where they will be broken down and turned into ATP, adenosine triphosphate, because that is the molecule that powers our body. Yes, we eat carbohydrates, we eat proteins, we eat nucleic acids, and we eat fats, but we cannot fuel our cells directly with those. They have to be converted first into ATP, adenosine triphosphate. And that's where your mitochondria come into play. Whether you're in a plant or in an animal, the mitochondria are going to convert those proteins, carbohydrates, and fats into ATP, adenosine triphosphate, which our bodies can now use. Okay, disaccharides, okay, these are basically two monosaccharides stuck together. And as we learned earlier, okay, the way we put two molecules together is what process? Think about it. Dehydration synthesis. Okay, and what does that mean? That means we have to take a water molecule out between the two monosaccharides. So, what is the formula for a simple disaccharide? Well, if each monosaccharide is C6H12O6, you might think that it's going to be C20 or C12H24O12. Got two monosaccharides put together, right? But don't forget dehydration synthesis. We have to take that water out. So actually, what's going to happen is we are going to take the two monosaccharides up here, take a water molecule out, and we're going to end up with a disaccharide with a formula of C12H22O11. And every time we add another monosaccharide, we have to take another water molecule out. Okay? So that's what happens. Polysaccharides, many sugars, can be multi-purpose. They can play a role in short-term energy storage, such as starch. This would be amylopectin and amylose are both starches. Glycogen is another storage form of sugar, and it's found in animals, where starches are found in plants. Okay? And there's some differences. The starch, the sugar molecules, the glucose monomers in starch and in glycogen are put together in what we call the alpha glucose pattern, whereas structural molecules, chitin, which makes up the exoskeleton of insects like a roach, and cellulose, which is what wood is made up, because cellulose is the sugar molecule, a polysaccharide that we find in the cell walls of plants. These are put together with a beta glucose. And you're like, what the heck's that? What's the difference there? Well, the difference is a slightly different structure, which leads to a slightly different function. So here you can see the alpha glucoses, and look how it's between a carbon number 1 and carbon number 4 of two adjacent glucose molecules. And we can see this oxygen's kind of down in this down position here, okay? But beta glucose monomers, they do slightly different. This one's up, this one's down, this one's up, the next one's down. Okay, we have enzymes in our bodies, and most animals do, that can break down starch, this bond. It can break this bond apart. Okay, we do not have enzymes in our body that can break this bond apart. We have to it's undigestible for us. Okay, it's what we call fiber. So like I say up here, the enzymes that digest starch cannot hydrolyze or break down the beta linkages in cellulose, so it passes through our gut system undigested. It plays a role in keeping us regular, if you will, and that's why you got to eat those fibers and stuff like that, because if you don't, then what will happen is that what we're seeing nowadays is that we're seeing a big increase, especially in younger adults, of things like colon cancer, which generally doesn't happen until you're my age. All right. So, but because of the poor diet and the lack of fiber in our diets, we're seeing this happen more and more in younger people. Okay. Now, a lot of organisms can digest the cellulose, but they can't really do it themselves. Instead, they have a symbiotic relationship. Symbiotic, sim means together, biotic means living together. So living together, have a symbiotic relationship with different types of microbes that do allow them to break down that cellulose. The cow itself or the termite doesn't break it down, but it's the microbes that live in their guts that break it down. Okay. So we store excess glucose in our blood in the form of glycogen in our muscle cells and in our liver cells. So after you've eaten a big lunch, your glucose levels go shooting way up in your blood. Your couple of receptors detect that high spike of glucose, and your pancreas releases insulin. Insulin is a protein that acts as a signal molecule that travels all through your body, and it causes the uptake of glucose from your bloodstream into your cells, especially your muscle cells and in your liver cells, and therefore it lowers the amount of blood sugar you have. Now, if it's been a few hours since you've eaten, your blood sugar levels decrease. Another hormone is released by the pancreas called glucagon, and that causes glycogen to start to hydrolyze. It starts to break the glycogen down into individual glucose molecules, which then get transported out to the blood and brings your glucose level back on up. So our bodies are maintaining this kind of like homeostasis, trying to, not too high of a sugar level and not too low of a sugar level. Okay, so that's why we do that. Now that gets us, there's carbohydrates. Now we're moving into fats, okay, lipids. All right, now lipids are really diverse. There's phospholipids, there's fats, there's waxes, there's oils, there's steroids, lots of different names and structures and functions for the different types of lipids. The one thing they all have in common, however, is that all lipids are hydrophobic, meaning they are insoluble in water, i.e., they don't mix in water, but they can be broken down and dissolved by non-polar substances. So if you have a fat, take gasoline, or grease and take gasoline, which is a non-polar substance, and it will dissolve that fat and break it down. Now, it's a little dangerous. You don't want to get near sparks when you're doing that. Hate to turn you into a human candle, but we don't, so don't do that. All right. That can be very dangerous. But if you take lipid molecules like, oil and throw it in some water, it's going to spontaneously cluster together in a wet environment in order to kind of exclude the water. So here's some diverse structures of lipids right here. The top one is a phospholipid. These are a very important class of fats, and they are unique because remember what I said earlier, all fats are hydrophobic. Okay, except for phospholipids. Now, phospholipids have a hydrophobic tail that consists of mostly all carbons and oxygens. There's a lot of carbons here. That's CH2 multiplied by 14 times. And here we have CH2 multiplied by 7. We have a double bonded right there. And then we have CH2 multiplied by 7. They're capped with a methyl group at the end. All right, but this portion of the phospholipid is hydrophilic, and phospholipids make up your cell membranes. Very, very important lipid. This is a triglyceride. This is which is composed of 3 fatty acid chains that are joined to a glycerol molecule. And then you have steroids. All steroids pretty much are derived from cholesterol, and they look the same, as we saw in our earlier presentation, this 4-ring structure. The only thing that's different about the different types of steroids is the functional groups. Here we have a hydroxyl group, an OH group. Here's a methyl group. Methyl groups make it non-polar. Another methyl group, all methyl groups. So this steroid, and this has one hydroxyl group, but it has so many more non-polar methyl groups that this molecule is insoluble in water. Okay, and the same goes for the terpene, and the same goes for triglycerol over here that we see. They're still insoluble in water, mostly non-polar. Now, a couple of fun facts. Fats are great at long-term energy storage because per unit weight, they have twice the amount of calories as do carbohydrates. And think about that. Plants don't move. They can store all their excess energy as starch, i.e. carbohydrates. Animals move. So we store our extra energy reserves, long-term energy reserves, as fat because that allows us to move around much more efficiently because it's much more dense energy storage. Now, the part of plants that do move, and you're like, what part of plant moves? Seeds move. Okay, they move by wind, they move by birds, they move by animals, they move by water. They move, sometimes extraordinarily long distances. And you think about a seed, the energy in seeds is mostly in the form of oils, i.e. fats. Okay, so the part of the plant that moves uses fats as an energy storage molecule. Okay, now I say you have saturated and unsaturated fats up here. You may have heard of those terms before, and we'll talk about what those basically are in a second. Well, fats are built just like other molecules. They're built through dehydration synthesis. Here we have a fatty acid. Why is it called a fatty acid? Well, it's all carbon hydrogen bonds. That makes it a fat. And fats are composed of carbon, hydrogen, and oxygen, but not in a 1 to 2 to 1 ratio like carbohydrates. In fats, there is a whole bunch of carbons, a whole bunch of hydrogens, and only a few oxygens. Like here we have 2 oxygens, and what? You got 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11. You got like 15 carbons there. Okay. Also, I want to point out the carbon hydrogen bond is energy rich. And when these bonds break, they release energy. That's why fats store so much energy. Okay, they're all carbon hydrogen bonds. Hydrocarbons, things like oil and gas and things of that nature, they are only carbon hydrogen bonds. And when all of those bonds are broken simultaneously, boom, and that's why hydrocarbons are explosive. Okay, just like a fat, you can set a fat on fire and it's going to burn. If your mom's grilling a steak on the grill and all of a sudden it catches on fire, that fat's going to burn like crazy. Okay. There's fatty acid. So saturated and unsaturated fats. On the left, we have a saturated fat. On the right, we have an unsaturated fat. Just by looking at them, you can see some differences. Saturated fat, long, straight tails. The unsaturated fat's kind of going all over the place. It's got wanky legs. Okay? Why? Because of double bonds. Double bonds put a kink in the hydrocarbon tail. Okay, and it causes those fats to not be able to pack in tightly. Whereas the saturated fats are saturated, they have the maximum saturation of the number of hydrogens that can be there. Okay, every single carbon is surrounded by hydrogens. Here, because there's a double bond, there's a hydrogen missing over here. There's another hydrogen missing here. So we got 1, 2, 3, 4, 5, 6 hydrogens are missing in this particular fatty acid compared to this one, which is saturated. Okay, so that gives animal fats and some tropical oils like palm oil and coconut oil, which are also saturated, some interesting properties. They are solid at room temperature. Think of butter or steak fat. Whereas plant oils and fish fats are unsaturated. They are liquid at room temperature, generally healthy versus unhealthy that we see in things like butter and steak fat and so on and so on. So the phospholipids, we'll spend a lot of time with phospholipids when we talk about the cell membrane, but you need to know what they are. Remember, these molecules are both hydrophobic, i.e. the fatty acid tails are hydrophobic, and hydrophilic, and that is the phosphate head. It's hydrophilic because of this negative charge here, which will be attracted to the polar water side. Okay? And these molecules are extremely, extremely important. And just recently, a paper was published saying that they found, I can't remember where it was, on a meteorite or on a comet sample or something like that, they found this part of this phosphate head. So again, kind of there's that panspermia hypothesis about the origins of these complex macromolecules. Okay? And so if you take phospholipids and you throw them into water, they will spontaneously form this thing we call a micelle or a micelle. And basically the hydrophilic phosphate heads will orient themselves towards the water side, and the hydrophobic tails will exclude the water on the inside. This would be like a sphere in the water. Now, that's a little bit different than a cell membrane. A cell membrane consists of a phospholipid bilayer. But above these hydrophilic heads, you would have the exterior part of the cell, which is in most cases is going to be a watery environment. And at the bottom here, you're going to have the interior part of the cell, which is also going to be basically a watery environment. So this is what makes up your cell membrane. The structure is going to lead to its function. The interior part of this phospholipid bilayer is completely hydrophobic, and that's going to prevent things like ions and large molecules and things that have a charge from actually moving through this membrane. But small things that are nonpolar can easily pass through the membrane. So this membrane is what you're going to hear the term selectively permeable. It's going to allow certain things in and keep other things out. Do you remember steroids consist of this four-ring structure, and the only thing that differs between the various steroids are the different functional groups that you see on it. Here's a methyl group, here's a methyl group, here's a hydroxyl group, here's a long hydrocarbon tail. And you might, what the heck is that thing? Well, there would be this is called a line model, and there's actually a carbon here, a carbon there, a carbon here, another carbon there, and a carbon here. And coming off of those carbons are going to be all the hydrogens that would be associated with that. It's just a simple, quick way of drawing molecules. And that is it for lecture 1.5 on carbohydrates and lipids. So hopefully this will help you with it. Alrighty, guys. Thank you for watching.